Yes, please. Yes, Dr. Golani.
Right, sir. Good evening to all. A warm and heartfelt welcome to each and every one of you joining us for today's enlightening webinar. I am Dr. Pradeep Golani, the co-convener of this event, and it brings me immense pleasure to witness such a diverse and engaged audience gathered virtually. Your presence not only enhances the vibrancy of our discussions, but also signifies a shared commitment to the pursuit of knowledge. I eagerly anticipate an insightful and collaborative session as we embark on this intellectual journey together. By consistently offering captivating webinars, SPSR has established itself as a leading platform for knowledge exchange and professional development in the pharmaceutical sciences. The audience greatly benefited from the webinar, gaining insights from the expertise shared by the eminent speakers. Thank you for being a part of this enriching experience. Today is a significant milestone as we gather for the 40th SPSR International Webinar on adopting genome editing technology in combating cancer disease. This event is a part of a distinguished series hosted by the Society of Pharmaceutical Sciences and Research under the esteemed mentorship of Dr. Shashi Alok, National President of SPSR. Under his guidance, the SPSR continues to play a pivotal role in advancing knowledge and innovation in the field of pharmaceutical sciences. The SPSR is dedicated to advancing the pharmacy profession through a multifaceted approach. Our objectives encompass the organization of conferences to campaign the causes of pharmacy, contributing to programs and services that address the health needs of India and focus on disease prevention. Additionally, we actively recognize and honor distinguished pharmacists who contribute significantly to the encouragement of the pharmacy profession. The core of our mission is the promotion and facilitation of the acquisition, dissemination, and exchange of pharmacy knowledge, ensuring a robust flow of information about the practice of pharmacy and pharmaceutical sciences. Through these objectives, we aim to elevate the standards and impact of the pharmacy profession, fostering a dynamic and collaborative environment. We are truly honored to welcome esteemed speaker Dr. Joseph Bamidele Minari, Associate Professor and Team Lead, Genome Editing Research Group, Department of Cell Biology and Genetics, University of Lagos, Akoka, Lagos, Nigeria. Dr. Joseph Bamidele Minari brings invaluable expertise to our event, and we eagerly anticipate gaining insights from his wealth of knowledge. His presence enriches our discussions and adds significant value to our exploration of latest advancements in the pharmaceutical sciences, particularly in the context of cancer chemoprevention. We are grateful for his participation and look forward to a truly enlightening session. In our esteemed gathering, we are honored to have distinguished presence of Mrs. Monika Sabharwal, the convener of this webinar, National Secretary of Society of Pharmaceutical Sciences and Research, and Managing Editor of IJPSR and IJP. Her multifaceted contribution and leadership in these capacities underscore her commitment to advancing the field of pharmaceutical sciences and research. We are privileged to have Mrs. Monika Sabharwal with us today, guiding our webinar with her expertise and dedication. Without further delay, I have the distinct pleasure of inviting Mrs. Monika Sabharwal to extend a warm welcome to our esteemed guest speaker, Dr. Joseph Bamidele Minari. Over to you, ma'am.
Thank you so much, Dr. Pradeep Gulani. Hello, everyone. I am Monika Sabharwal, the convener of this esteemed webinar, and it brings me immense pleasure to welcome you all to the 40th SPSR International Webinar on adopting genome editing technology in combating cancer disease, organized by Society of Pharmaceutical Sciences and Research. The SPSR stands as a beacon of knowledge and collaboration in the realm of pharmaceutical sciences with a steady commitment to advancing research, education, and innovation, SPSR has continuously elevated the standards of pharmaceutical community. Through its tireless efforts and dedication, SPSR has created a platform for pharmaceuticals researchers to exchange ideas, share discoveries, and foster meaningful connections. On the hallmark of initiatives, the SPSR is its series of webinars, which have emerged as invaluable resources for the global pharmaceutical community. These webinars serve as a dynamic forums where experts from diverse backgrounds converge to explore cut-edging topics, address pressing challenges, and chart the course for the future of pharmaceutical research and development. The seamless organization and execution of these webinars by the SPSR have garnered widespread acclaim, attracting participants from across the globe. 40th SPSR International Webinar titled Adopting Genome Editing Technology in Combating Cancer Disease stands at forefront of cutting-edge medical research and innovation. Cancer continues to pose a significant challenge to the global public health, with millions of lives affected by its devastating impact every year. However, advancements in the genome editing technology offer a glimmer or hope in the fight against this formidable adversary. Today, we have privilege of delving into the potential of genome editing to revolutionize cancer treatment strategies, offering personalized and targeted approaches that hold a promise for improved patient outcomes. I'm truly honored to introduce our distinguished guest today for today's webinar, Dr. Joseph Minari. Dr. Minari serves as an associate professor and the team lead of Genome Editing Research Group at esteemed Department of Biology and Genetics, University of Lagos, Nigeria. With the career marked by the exemplary expertise and a groundbreaking contribution to the field of genome editing, Dr. Minari stands as a beacon of innovation and excellence. His pioneering research endeavors have not only expanded our understanding of genomic editing technologies, but also have paved a way for transformating advancements in combating disease, particularly cancer. Today, we are privileged to have Dr. Minari to share his wealth of insights and experience with us, offering invaluable perspectives that promises to inspire and enlighten all of us. It is my immense pleasure to extend a heartfelt welcome to Dr. Shashi Alu, the esteemed National President of SPSR. Dr. Shashi Alu's exceptional leadership and unwavering commitment to the scientific research have played an important role in propelling our community forward and fostering the culture of innovation. His visionary guidance has been instrumental in orchestrating a remarkable transformation within the SPSR, positioning it as a beacon of excellence in the field of pharmaceutical sciences. I would also like to express my sincere appreciation for the invaluable contribution of Dr. Pradeep Golani, the co-convener of this webinar, and the organizing committee members, Dr. Payal Mittal, Dr. Saurabh Kose, Dr. Sanjay Nagdev, Mr. Angar Agarwal, and Mr. Junaid Tantre. Their tireless efforts have played a crucial role in transforming the vision of this event into a reality. Behind the scenes, there is an exceptional team who has been a backbone for this seamless organization. Their tireless dedication and commitment to the excellence have always been driving force behind the success of this event. I extend my sincere gratitude to each and every SPSR team member for their meticulous attention to the every detail, which have made this webinar possible. The overwhelming response to this webinar is truly commendable. We have received more than 2,500 registrations from participants spanning over 15 countries. highlighting the global significance and the collaborative spirit of shared commitment to combat cancer. As we embark on this enlightening journey together, let us embrace the opportunity to learn, share, and grow together. May this webinar serves as a catalyst for positive transformation in lives, both personally and professionally. Once again, thank you for being part of the 40th SPSR International Webinar. And without further ado, let us embark on this enriching exploration into adopting genome editing technology in combating cancer disease. Our distinguished speaker, Dr. Joseph Minari, awaits us to enlighten us with invaluable insights. Let us delve into this journey of discovery together. And do not forget to subscribe our SPSR YouTube channel to stay updated on the latest webinars and research insights. Thank you. And let's begin the 40th SPSR International Webinar. Over to you, Dr. Pradeep Golani.
Thank you, Mrs. Monika Sabharwal, ma'am, for allowing me to conduct this webinar. And now let's begin with a brief introduction of Dr. Joseph Bamidele Minari. So, Dr. Minari is an accomplished associate professor at the Faculty of Science, University of Lagos, Nigeria, with a rich academic and professional background spanning over 2 decades. He specializes in the cell and molecular biology, genome editing, and DNA forensics. Dr. Minari began his academic journey at Joseph Ayo Babalola University before joining the University of Lagos. Throughout his career, Dr. Minari has demonstrated exceptional mentorship, supervising over 40 students at both undergraduate and postgraduate levels. His research contributions are significant, with over 70 publications in esteemed journals, both domestically and internationally, with ongoing publications as well. He is actively involved in various professional and academic groups, holding positions such as National Vice President of Nigerian Society for Experimental Biology and National Secretary of Society for Forensic and Analytical Scientists, Nigeria. His memberships include prestigious organizations like the Biochemical Society of United Kingdom and the American Society for Biochemistry and Molecular Biology. Dr. Minari is also committed to continuous learning and development, participating in workshops and training sessions worldwide. Notable among these are his involvement as a facilitator in DNA molecular biology training workshops and his participation in genome editing and gene drive courses. His research endeavors have been supported by numerous grants, including awards from the University of Lagos, United Kingdom, Research and Innovation, Lagos State Government, and Tedford Federal Government of Nigeria. Dr. Minari's success in securing and managing multiple grants underscores his stature as a distinguished researcher and with profound contributions to the field of cell and molecular biology, genome editing, and DNA forensics. The grants he has obtained, including the University of Lagos CRC grant, UK GC FR COVID-19 grant, LESTRIC grant for developing a SARS-CoV-2 vaccine, and TEDFund grant signify the recognition and support he has garnered from the prestigious institutions and government bodies. These grants not only attest to the significance and relevance of Dr. Minari's research, but also highlights his capability to address pressing scientific challenges such as COVID-19 pandemic through innovative approaches. With each grant, Dr. Minari has demonstrated his adeptness in securing funding and managing research projects of substantial importance, showcasing his leadership in driving impactful scientific advancement both nationally and internationally. Dr. Minari's contribution extends beyond research to the dissemination of knowledge through conference presentations. He has been a speaker and moderator at various conferences, both locally and internationally, covering topics ranging from biochemistry to forensic sciences. Noteworthy conferences include those hosted by the University of Cape Town, University of Bristol, University of Warwick, and Humboldt College International Conference. Overall, Dr. Minari's career is marked by a dedication to advancing scientific knowledge, fostering academic growth, and making impactful contribution to his field, both in Nigeria and on the global stage. It brings me immense joy and pride to share that we have garnered the participation of over 2,500 registered attendees from across the globe, representing a diverse array of countries. Among them, we have significant presence from India, Nigeria, Ghana, Iraq, Ireland, Kuwait, Nepal, North Macedonia, Pakistan, Philippines, Sri Lanka, Ethiopia, Indonesia, UAE, and United Kingdom. The overwhelming response from such a wide-reaching and varied audience is truly humbling. We extend our heartfelt gratitude to each and every participant for choosing to be a part of this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed guest speaker, Dr. Joseph Bamidele Minari with us. Welcome, sir. Kindly, I request you to share your PowerPoint presentation and talk.
Thank you very much for having me, and such a great pleasure to be given the opportunity to speak on this platform. And I'm very sure that at the end of the day, we'll be able to rub minds together, share ideas, and then be able to move science forward and be able to get one or two things that we can actually use to combat the issue of cancer, which has actually bedeviled the whole world. Okay, so let's go straight to the matter of the day that we want to discuss. I want to talk about adopting a genome editing technology in combating cancer disease. Like we've had a associate professor from University of Lagos, and we have a research group here called the Genome Editing Research Group, and which I happen to be the team lead from Department of Cell Biology and Genetics. So let's start off. What is cancer? Cancer is a disease that has been known to be majorly characterized by the uncontrolled division of the cells. In the world of science, we know that cells are supposed to replicate and they are supposed to be controlled. But when you have uncontrolled proliferation of the cells, it accumulates to form tumors. And most of the time, we have the cause of this as being the genetic disease that actually emanates and show forth at the cellular level. We have far over 100 kinds of human cancer that has been known. And most of the time we have different types, different classification, depending on where the cancer is emanating. So when we look at this, we have this normal cell division, okay, that's supposed to go probably for growth or probably to maintain some of the worn out cells in the body. So the cell supposed to go this way, and if there are damages or there are issues with the cells supposed to probably engage in a suicide mission called apoptosis, or take care of the cells while the normal cells actually continues the journey. But we have in cases like this where we have cancerous cells, we have damages, and then it moves on to the next. So the damage that is being caused probably due to mutation, first mutation going to the second mutation, to the third mutation, to the fourth, and then you have accumulation of those mutations, and that leads to some of these cells aggregating and then forming a mass of tumor. Now we have different categories of cancer. So let's just look at some of the major ones that we have. We have the category called the carcinoma. The carcinoma cancers are those cancers that originate from the skin or the tissues. Okay, so most of the time, they line or cover the internal organs. So these ones are referred to as the carcinomas. Then we also have the sarcoma. The sarcomas are the kind of cancer that actually originates from the bone or the cartilage or the fat or the muscles or the blood vessels or the connective or supportive tissues. So all these cancers that originate around this region is actually referred to as the sarcoma. And then we have the leukemia. These are the cancer that starts in the blood, forming tissues such as the bone marrow. You know, the bone marrow being the source of major production site of the blood. So when we have a cancer that emanates from the bone marrow, it is referred to as the leukemia. And then we have the lymphoma and the myeloma. This cancer begins from the immune system. So when we have cancer that also originate from the immune system, they are classified.
Hello, Dr. Minari. I think your sound is not audible and your PPT is not visible.
Hello, can you hear me?
Yes, sir. Now it's audible also and visible.
Oh, okay. Thank you.
You may continue.
So sorry.
Right.
Yes, sorry for the break in transmission. There was a network problem from here. So I was talking about different categories of cancer. I talked about the carcinoma, the sarcoma, the leukemia, the lymphoma, and the myeloma. And then we also have the central nervous system cancer. These are the cancer that actually begins from the brain or from the spinal cord. Now, we have these principles of carcinogenesis, really. When we look at it at the genetic level, we discover that neoplastic transformation actually is progressive, and we actually have accumulation of it before it can actually degenerate, just like what we saw earlier in that picture, that sometimes you might have that level of mutation from 1 to 2, and then you have in multiple hits before genetic changes can actually occur. And the alteration could come in various ways. It could be that there is a gain of function from the proto-oncogenes that's supposed to signal proliferation of the cells. When they lose control and there is a gain of function of those genes, they become oncogenes. This can actually trigger the possibility of carcinogenesis. Also, we also have the tumor suppressor genes. These are genes that are actually supposed to inhibit If at all there is proliferation, there's stimulatory genes are failed and they are just opened up, there should be a regulatory gene at this point. So the tumor suppressor genes actually supposed to hold them. So if we have loss of function of these tumor suppressor genes, it can actually lead to the proliferation of cells. We can also have gene regulation apoptosis failure. Sometimes when there are damages, if it doesn't go the way of regulation, the normal cells that have been damaged are supposed to be sentenced to apoptosis, where they are actually being destroyed. And then we also have the DNA repair genes during the cell cycle phase, at the S phase, where we have replication of the gene. So if the genes there also fails to repair, it can also degenerate to us having uncontrolled division or proliferation of the cells. Now we have characteristics of cancerous cells. After that failure has occurred, what happened to the cells? We can have because the signal has actually failed, so you have a sustained proliferative signaling. So the proto-oncogenes, they fail, they are now oncogenes, and that can just lead to the cells just continue to multiply. And then we also have evading growth suppressors, just like we said for the tumor suppressor gene. So this is another characteristic of the cell, cancerous cells that we just have. So they're already evading the tumor suppressor genes, because the tumor suppressor genes has lost their functions, and so their proteins will not be able to perform their function well. Then we also have activation of the invasion or metastasis. So they can they can evade other cells, they can go far away and then start up the growth there. Those are part of the characteristics of the cells. And then enabling replicative immortality during the S phase. They've lost that repair, and so it just makes them to continue to replicate. We also have induction of angiogenesis. Okay, so we have the blood vessels also being amplified to supply the proliferation of the cells the kind of energy that they want for them to continue. And we also have resisting cell death, which is supposed to be the apoptosis, where they're supposed to take care of the damaged cells. We have different risk factors of cancer. We have the genetics, we have the family history, we have the age, we have the lifestyle, we have environmental factor, we have diet, we have drugs, and then we have infections. These are some of the risk factors that can actually prone someone to having cancer. Now we have the burden of this cancer. I'm very glad that during the introduction, it was also mentioned about the burden of the cancer. Let's look at it even from 2012. A very good statistics I was able to take in 2012. You will agree with me that cancer has been a leading cause of morbidity and mortality, even up to date. In 2012, approximately, we had about 14 million new cases. And then it was even projected then that new cases expected to rise by 70%. We're going to look at it shortly, maybe in the next slide. Let's see what it has actually got into. And cancer is the second leading death globally. And about 8.8 million died in 2015 just because of cancer. So globally, invariably, you can see 1 out of every 6 deaths is due to cancer. So that tells you the enormity of the burden of cancer. And that is the reason why all hands must be on deck to ensure that we are able to actually combat this cancer and fight it. And approximately 70% of death from cancer occur in low and middle income countries, just as we have in the statistics. Now look at it at around 2020. The new cases we have for females alone, we have breast cancer leading the pack with 24.5%, followed by colorectal cancer, followed by lung cancer, 8.4%, colorectal is 9.4%, and the cervix uteri 6.5%, the thyroid 4.9%, the corpus uteri 4.5%, stomach cancer 4.5%, and different other little cancers here and there that if you begin to create a space for them, it will be too numerous. Classify them together, they stand about 37.8%. And the total for female cancer estimated for 2020 was over 9,227,408. That's for females only. Now, in this same 2020, for males, we have the lung cancer taking the lead in 2020 of 14.3%, prostate cancer 14.1%, colorectal cancer, colorectal cancer 10.6%, stomach 7.1%, liver 6.3%, bladder 4.4%, esophagus 4.2%, and every other one stands at 39.1%. And we have a total of over 10 million new cases of cancer. So put together, that is about 19 million going to 20 million new cases in 2020. So you can see that the number is actually increasing when we have probably about 14 million in 2012 and in 2020, just about 8 years apart, it has increased. So you can imagine what we'll be having now in 2024. So definitely the number has increased. Now let me localize it a little bit. I'm from Nigeria, and statistics, even as at 2021, Nigeria is a country of a population of about 250 million people, and the cancer death has been estimated, especially the reported ones, about 78,809 people have been reported dead on cancer, and new cases reported in 2021 is about 124,815. Breast cancer is the one taking the lead with about 37%. So you can see the scourge of cancer everywhere, the burden of cancer everywhere, globally and even locally. I know if you check all over the world, countries like India, countries like Kenya, and countries like Ethiopia, Indonesia, and so many other countries, I'm very sure that the story will be virtually the same or very similar in terms of the death that cancer is actually causing. And that also is very, very instructive for us to know that as scientists, we actually need to move towards finding how to stop the scourge of cancer or to reduce it drastically. Yes, there are valuable treatment and management that has been used over the years to manage or to treat cancer. We have surgery, we have chemotherapy, we have radiation therapy, we have a targeted therapy, we have immunotherapy, we have a hyperthermia. Hyperthermia is when they actually apply heat to ensure that maybe they're able to curtail the spread of the cells. We have the stem cell transplant. We have the photodynamic therapy. And, you know, this application of light, likewise the laser was also application of light to see how they can use light to kill some of these cells. We have blood product donation and transfusion. Also, in some part of the world, even like Nigeria, we have the complementary and alternative medicine. In this part of the world, like places like Nigeria, I know they believe, quite a number of people believe in this one and such, where we have dietary supplements, herbs, vitamins, and minerals. You know, when people, when they are down, probably sometimes because of the cost, even cost of going for surgery, the fear of going for surgery, and some other side effect that is associated with surgery, chemotherapy, radiation, and all other therapy that have been outlined above, people resort to supplements, herbs, vitamins, and minerals, using them to see how far it can go. But the truth is that sometimes some of these things does not really stop it. In some cases, it might probably reduce the proliferation a bit, or in some cases, if it is for chemo prevention, might delay the onset. Because as long as if it is the kind of cancer that actually emanated from the genes, and the genes have been treated, altered, it's most likely that it's just a matter of time. It will still come up. And so the alternative, yes, is good to just ameliorate or to manage the person, maybe pending the time that the person is going to spend. But does it really care? Does it really cure it? Does it really completely take care of it? We also have what we call the palliative or the supportive care. So in this the focus is mainly on patients to get relief, on some symptoms or effect that comes with it. Maybe somebody feeling fatigue, somebody feeling pain, nausea, and so some of these shortness of breath. So some of these symptoms can be treated on their own, but that does not still stop the cancer itself, because the cancer itself will just continue proliferation, but we're proliferating. But some of the side effect that comes with it might actually be managed to reduce the pain, to reduce the fatigue, and then the person will actually be constantly be on drugs to make sure that they are able to manage some of these things that comes with the care of the cancer patients. Now, let's look at this. So when we talk about the cancer patient, we talk about the therapy. Now, one thing that we can see is that, yes, let's take two of the kinds of therapy that we mentioned earlier. The radiation therapy, let's look at the chemotherapy as well, okay? That is sent towards the cancerous cells. The pink color represents the normal cell, and the darker color represents the cancerous cells. So for radiation, we try to use radiation to deaden some of these tumors, some of these cancerous cells. But the question is, will that stop the gene that has lost the signal of a tumor suppression, or will it stop the oncogene that has gained function to continue to proliferate, or will it affect the apoptotic pathway that have also lost its function, and so on and so forth? So it will deaden the cells, but over time, some of those growths might still continue. And also for the chemotherapy, even for the surgery, when you remove those cells, but the genes are still in those cells, definitely the growth might actually continue to come up. Okay, so, and one of the major setback in some traditional cancer therapy that we have, such as the chemotherapy and radiation therapy, is the inability of the drugs that you are using on the radiation to actually distinguish between the malignant and the healthy cells. So as you are sending the drugs into it, the drugs will not differentiate between the malignant cells and the healthy cells. So as it's destroying the malignant cells, it's also destroying the healthy cells. So both cells are destroyed during the therapy, so resulting most times to undesirable side effects and even collateral damage of the normal cells. And in some cases, eventually, the person being treated might lose his life, because if the tumor is around a vital organ, and then you are trying to maybe close to the vital organ, and then you are trying to destroy some of the malignant cells, and at the same time, because your chemotherapy is not actually targeted exactly on those cells, it will destroy some of those cells, even radiation therapy, and some of the cells of the vital organs might also be destroyed, and then the vital organ might be impaired. And when the vital organ is impaired, that can actually lead to the death of the person involved. So this is a major problem that is being faced when using the conventional cancer treatment that we have. And that is why up to date is still a major problem that in treating, in adopting the conventional cancer therapy to treat or to manage the cancer patients. Now, here we are. But in genome editing, in contrast, it combats cancer by targeted therapy. So only cancer cells, that means those cells that are not healthy, will be targeted and will be removed, will be eliminated. So that is what genome editing brings on board. So rather than using chemotherapy, radiation therapy, or other forms of therapy that just targets both the healthy and the malignant cells, you can edit your genome in such a way that it will target at your desired unhealthy cells. And that's the major reason why we are giving this talk today, that how can we now adopt this, because it's very, very important that all hands must be on deck for us to go towards the genome editing aspect technology to see how we can adopt it, apply it in combating cancer. So let's start. Genome editing. What is genome editing? Genome editing is a type of genetic engineering which targets the sequence in the genome. And it does this by, you can insert, you can delete, or you can replace a portion of the genome or the segment of the genome by using a particular molecular scissors. So the way it works is that you can just imagine a normal scissors that you are using it to cut, even though it doesn't work like that literally, but eventually what you get from that is what looks like it. So you are looking at a portion here. Probably this is a sequence that you are not interested in. You are able to cut this particular sequence, and then you might leave it like that. That is deletion. Okay, you might decide to insert, okay, or you might even cut somewhere else to come and paste there, just like the way you do literally, but you can do it in that way, not the same procedure of cut and paste physically, but that is exactly what happens. We have different mechanism of genome editing. We have the meganucleases, we have the zinc finger DNA binding domain, we have the darlings. You know, these are various methods you can use for genome editing. But however, in recent time, the CRISPR-Cas9 system has actually gained ground in effecting the kind of editing that we actually want. And it happens because it's actually easy to engineer and multiplex, unlike others that's a little bit difficult to engineer. And it also have some level of specificity a little bit higher than others. So when we talk about the CRISPR-Cas system, so the CRISPR itself means the clustered regularly interspace short palindromic repeat, and is actually a mechanism that a bacteria actually use to defend themselves. So what they do is that most of the time when they have an attack from the virus, okay, from the bacteriophage, when it comes, they are able to secrete their Cas to digest the virus, and immediately they do that, they now store a memory, a portion of that virus in their system. So it serves like an immune system for them. Okay, that's what they use to do. So it is that cluster that you have, that is where CRISPR actually came from, and it has been an important tool that goes beyond the bacteria, and that's what we are using now. And what does it do? So we're able to get the guide RNA, and then you have the CRISPR with it, and then you have the PAM site, the PAM sequence that actually helps you to direct and to lead the guide RNA to know from which sequence that the Cas is actually going to cut. So this guide RNA system has actually helped us to give us some high level of specificity, because of course, the guide RNA must have a particular location that he needs to go. So you must design your guide RNA in such a way that it has a target. So it's not just going to cut anywhere. So to some extent, you can have some level of specificity. And then from there, reading from the PAM sequence, the Cas then can now cut. And when you do this, you can actually cut. When you cut, you have some possibilities of what can happen. When you cut, when you leave it without inserting, it's possible that the portion, the sequence that you cut can actually join together using the normal double-stranded repair mechanism that happens with sequences. Call it non-homologous end joining. So you can make it to join, or you can actually have a template, okay, on it, where you also have a complementary strand to it, which is called homology-directed repair. So in this one, you actually need the template. So your cutting can lead to deletion, to be that you want to delete a particular portion, which can make it probably you want to knock out a gene, or it's possible that you are actually inserting a particular portion. Also, it could also be that you want to cut and paste. There's a particular kind of sequence, a particular kind of expression that you want to cut and paste on a particular portion. It's also possible that you are able to do that. Okay, so let's talk about the CRISPR-Cas9 and cancer therapy now. Okay, so we know now what the CRISPR can do as a genome editing tool. So what it can do is that it can actually target a particular sequence in the genome of interest for you, and then you can actually edit those genome either to gain function or to lose function, as the case may be. These are some of the genes that we have that have been implicated in several cancer cases, and then a lot of them you can actually use your CRISPR-Cas to actually target and edit, probably to knock out, probably to disrupt, or you have a specific sequence you want to actually insert to actually give you a particular trait. Now let's look at the typical example. We have quite a number that if we begin to take them one after the other today, it's going to take us a long period of time, but I will use one as an example. Let's look at the T cells, for example. We have the T cells. So normally in the T cells, you can actually use the CRISPR to engineer the T cells, and it will make them to actually gain a receptor site. Now, I will explain how that works shortly. Now, normally the T cells in the body, when there is a tumor, the T cells recognizes the tumor and then supposed to attack the tumor and then get rid of the tumor. Okay, so you have the TCR gene, that expresses the receptor on the T cells. So the T cells recognizes the tumor cells and then is able to destroy it. But in the body, the immune system also have a checkpoint so that the T cells will also not be attacking its own cells. So there is a checkpoint. So that checkpoint will actually inactivate the T cells. Now, when you have a cancerous cells, when you have a patient that is developing, that's having cancerous cells, and the cells are multiplying, okay, so the checkpoint sees it as if there is a need to actually stop the T cells of not attacking itself because of the rate of proliferation. So when this happens, the T cells will not be able to actually destroy the cancer cells anymore because of the immune checkpoint. So because of the immune checkpoint, so the immune checkpoint are activated, okay, by binding to a receptor on the surface of the T cells, okay, to their ligands. So because of that, they will not be able to attack the T cells. So normally what is happening is that sometimes we can insert, using CRISPR, you can insert the gene, the CAR gene, which is the chimeral antigen receptor. If you insert into the T cells will develop receptor on its own. So it will develop receptor on its own. So at this point, it's now called the CAR T cells. So using the CRISPR, you edit those genes, insert the template of that gene, and then the T cells develop this receptor. So, and it's receptor of choice. So this receptor will be the receptor that will be able to recognize the tumor cells, so that when the tumor cells, when the T cell sees the tumor cells now, it will be able to destroy the tumor cells. So you are not relying on the normal T cells that the body is generating anymore, because this one cannot cannot destroy the tumor cells anymore. And why? This is the reason now. Now, if you look at these T cells, this T cell, on the surface, it has the PD-1 receptor. So this PD-1 can actually bind to the PDL1. And most tumor cells have been known to express PDL1. The expression of PDL1 is so high on tumor cells. So when it comes to this, PDL1 binds to the PD-1 on the T cells, thereby inactivating the T cells. So this definitely will not make the normal T cells to work. And that is why even sometimes when you engineer the T cells to have the CAR receptor, the TARO T cells, so when you have the CARO T cells, it might still not work because of the PDL. So in the CRISPR technology, what you now do also is that you need to knock out the PD-1 gene. So you need to knock out the PD-1 gene. So when you knock out the PD-1 gene, okay, you knock out all those genes that actually assist in the production of the normal T cells receptor, and also the normal receptor itself, okay? Because when it sees this, the tumor cells will be able to bind to it. So you knock out those genes, and then with your CAR T cells, you have conferred another. So this is TAR T cells here. You'll be able to confer another receptor site on the T cells. So the T cells will see the tumor receptor cells, and then you'll be able to destroy them. So meanwhile, you have taken care of all other receptor cells on the normal T cells. So this is what genome editing can do, and it works this way. You remove the blood from the patient. When you get the blood from the patient, you use your CRISPR-Cas to edit the T cells in the laboratory. Okay, so when you do that, you ensure that you have inserted the CAR into the T cells, so that the T cells can gain different receptors on it. Okay, and so these receptors is going to bind on the tumor cells. So not only that, you also ensure that all the genes that can actually confer attachment to the normal tumor cells are being knocked out. So you delete them. You delete those genes. So when you delete those genes, the T cells with the new receptor it has gained, you now be infused back into the patient, and then the T cells can bind to the tumor cells, and the T cells can now do its normal job. The CRISPR T cells bind to the cancer cells and then kill them. So this is one major area that work is seriously ongoing, and a lot is being done in these cells to be able to tackle and to combat cancer. Cancer is not a monogenic disease. Like, for example, the sickle cell disorder that recently the CRISPR has been able to tackle that, and there's some countries has actually adopted it. That is monogenic, so it's a little bit straightforward, that you just target the stem cells from the bone marrow, then you start it back, and then the expression, then the patient, the sickle cell patient will be fine. So this is most cancer could be polygenic, but we are now in this instance, we are taking advantage of the immune system that normally supposed to attack any form of cancer that is coming. So your target will now be targeting those T cells, re-engineering the T cells, conferring upon them another receptor that will be able to bind to the tumor cells or the cancerous cells, and then be able to destroy them. So from there, we are able to actually destroy those cancer cells, and any other genes that can actually confer the regular receptor or reception on the T cells will be knocked out using same CRISPR to delete the portion of those genes, or you can disrupt them by inserting a portion of the sequence that you are sure will be able to inactivate or knock out the gene that you are not interested in. Okay, so what are we doing in our laboratory in University of Lagos here, Nigeria? In our genome editing lab, we have two subsets, two sub-area. One part of us is working on the CRISPR-Cas9 edited microorganism for the production of industrial enzymes. We are working on that, and also we also have this aspect of the CRISPR-Cas9 edited cancer genes to improve efficacy of the available drugs. So we can see that we have two aspects of it. And one aspect, for example, the CRISPR-Cas edited microorganism for production, we've done quite a number of work along that line, where we have safe and environmentally friendly microorganism. We actually edited the microorganism, and the edited microorganism acted on the waste, and then we're able to increase the production of industrial enzymes that are good for commercial purposes. So what we actually did, that we got eco-friendly E. coli, and then we targeted one of the genes there, and then we were able to knock out the gene. So after knocking out the gene, we acted it on a waste that we got, and then we discovered that there was mass production of some of the enzymes that were actually tested, which was actually characterized. And these enzymes are industrial enzymes that are actually useful. So that's one aspect of our work in our genome editing lab. Then the work that is still ongoing is on we using CRISPR to enhance the efficacy of cancer drugs. Like I said earlier, there are so many genes that we can target. We're actually targeting one of these genes now in our lab, and then we want to see how we can actually disrupt some of these genes using the CRISPR technology as a genome editing tool to ensure that drugs that are used will be very effective, in combating the tumor cells, the cancer cells. So a lot of work is ongoing in our laboratory, and we hope that we are able to get good results. Now, why should we adopt genome editing technology in combating cancer? Number one, it's going to help to reduce mortality and morbidity associated with cancer. Of course, with all the available treatment that we have lately, we agree with me that the rate of mortality and morbidity is still very high. And so embracing the genome editing technology will definitely help us to combat this real time, and to be able to drastically bring those cancer down. Then number two, it will help to correct the genetic disorders. So if it's actually a genetic disorder, it might be difficult to treat, but by applying genome editing, you might be able to correct some of the possible alterations that we have on the sequence of the genome, and that can actually help probably in the expression of the gene and the translation to produce the appropriate gene that's supposed to be there to produce the right protein that we have there. Then number three, it will reduce side effects associated with these conventional drugs that we have. So, of course, we know, like I told you, there are a lot of side effects that comes with some of the conventional treatment, like we have the alopecia, where we have loss of hair. We have even eventually some people still losing their life after some of these conventional treatment, and a whole lot of other side effects that she's with it. But with the use of genome editing technology, which has been adjudged to be safe in the ones we have done so far, following the protocol, it will help largely to actually reduce the side effect that has been associated with it. Then number 3, another reason why we should adopt genome editing in combating cancer is that it will promote precision medicine. Lately now, everybody has been talking about precision medicine. Medicine wants to go towards actually tailoring the treatment actually to individual. And if there is one disease that actually needs precision medicine, it's cancer, because the cause might come from different angles. And like I told you, that we have multiple sources and multiple failure that can actually happen. So maybe from the tumor suppressor gene, or from the DNA replication, DNA damage repair system during the apoptosis, and so on and so forth. So knowing exactly where the cancer is coming from, and knowing the genes, those genes that are failed for a particular individual, you can actually get the stem cell from that particular person, and then we're able to correct it using the genome editing tool, probably using a CRISPR, which is we use in our lab to correct it and then get it back. So this will actually help in precision medicine. So not just general medicine of one fix all, just take one drug and then you give it, and which in some cases might not be okay for some people, and some might even have side effects on some people. But if it is what is being tailored directly to that particular individual, can actually help. And this can be done when we encourage and adopt the use of genome editing. Yes, we have some challenges that has been affecting us, especially in Nigeria as a case study. We still have poor awareness in Nigeria on genome editing. We have a lot of issues, even some ethical issues that goes with it. Traditional issues. Some people will believe, oh, why do you want to alter the genome? Why do you want to change the genome? Oh, is it not bad for you to change the genome? What if you get undesirable effects? So we have limited knowledge when it comes to genome editing, and then poor awareness. People don't even know what it is. They've been hearing it, but when we talk about genome editing, people just think, oh, it's just getting one chemical and just punching into something, but doesn't know that it goes with a lot of precaution, goes with a lot of safety guidelines, goes with a lot of ethical guidelines that you have to follow before anything genome editing product can actually be rolled out. So poor awareness we have in Nigeria, and also I believe in some other parts of the world, for those that are listening to me. Then we have also in this part of the world, we also have a challenge of sometimes endless waiting for some consumables. Sometimes when we place orders on some consumables, we have to wait endlessly. We have to be mailing, checking. So these are some of the challenges that we face in this part of the world. Then we have poor funding for research. Genome editing research is capital intensive because of the ethical issues that actually surrounds it, and the funds are not being pumped into it as required, because we have to take extra care in what we do. So normally the funds supposed to be available so that we will be able to conduct our research and then do it accordingly the way it should be done. Then inability to access collaborators is a major problem as well, because if we get collaborators outside, we might be able to pull resources together, maybe from India, maybe from Sri Lanka, from Ethiopia, from Ghana, from Kenya, from Indonesia, from Afghanistan, and other countries. If you're able to get collaborators, it will help a great deal. Some facilities that you have that we don't have might be able to work together in synergy and be able to get a very good result. So these are some of the challenges that we are having. Okay, this is me there standing, and there's some of my postgraduate students when we're having one of the practical sessions of genome editing, that we're trying to edit some genes in the lab. Actually, the result was successful, and we got a very good result. We're able to edit the gene. This is outside the lab, actually, in the main lab that we're trying to get some things done before moving inside. Okay, so in conclusion, I want to say that more attention and funding needs to be channeled into adopting genome editing in cancer management and treatment. Thank you very much for listening. Thank you very much for having me.
Thank you so much, Dr. Minari, for such a wonderful session on adopting genome editing technology in combating cancer. You introduced about various types of cancer and characteristics of cancer cells. You also educated us about the world scenario of mortality due to cancer in males and females with figures. You discussed in detail about CRISPR Cas9 gene editing technology, its advantages and limitations. You also elaborated on how you are working in laboratories with CRISPR Cas9 editing in microorganisms for production of enzymes and editing cancer genes to improve efficacy of available drugs. Thank you so much, sir. So there are some questions from the participants regarding gene editing technology for cancer treatment. So one of the participants has asked this question. Can you provide the examples of how genome editing has been utilized in preclinical, clinical, and clinical settings to target cancer cells?
Yes, that's part of what I showed. Let me project it. A lot of work is being done on the T cells. A lot of work is done at preclinical level. Some of them have been tried at a clinical level as well. Okay, T cells are being engineered. Okay, T cells are being engineered. I told you that T cells normally in the body supposed to attack cancer cells. Okay, but now they are being engineered to acquire these chimeric antigen receptor CAT. So what we have there is that this is a practical example of them getting the blood sample from the patient, now engineering the T cells to acquire artificial receptor sites. Okay, so this artificial receptor site will identify cancer cells. And then all other genes that can actually make the T cells to show the regular features will be knocked out, will be deleted. Okay, so they will now mass produce those T cells. And then you can see this one like the sac of the drip in the hospital. So you infuse this one back to the body of the patient. And so when the T cells get to the body, the engineered T cells will just, anywhere he sees those tumor cells, he will attack them, and then it will destroy those cancer cells. So quite a preclinical work is ongoing, and in some cases, some clinical work is also ongoing along this line. Thank you.
Right, sir. Right, sir. Thank you. Thank you so much. The next one is, sir, how does genome editing enable the development of personalized cancer therapies tailored to individual patients?
So how does it help to tailor into cancer patients? So like I said initially, you know, when it comes to the issue of cancer, different proteins, actually, when you look at proteomics, different proteins failure of different proteins can lead to almost the same cancer. So when you, okay, let me take, for example, breast cancer, for example. You know, in breast cancer, we have different types of breast cancer. We have the HER2 breast cancer. We have the triple negative breast cancer. So when somebody presents itself for breast cancer, and then you are just treating one breast cancer. So if it is triple negative breast cancer, and you are treating HER2 or RBB2 breast cancer, the drug is not going to function. So if for genome editing, you are actually editing the receptors of HER2, okay, so it's not going to work. Right. So, but if it is triple negative, is another kind of breast cancer. So you will take your sample and then you edit the gene based on the kind of cancer that person is, specific kind of cancer that person is having, and not just on general basis on say, okay, it presented on breast cancer, whether it's Lumina A, whether it's Lumina B, whether it's triple negative, you are just using the same thing. So you tailor it, get the gene, get the sample from the patient, engineer what exactly you want to do and the target of that particular gene that that is of interest to you.
Thank you, sir. You answered very well. The next question is, sir, what are the current strategies for delivering gene editing tools to cancer cells in vivo and how effective they are?
Okay, we have different strategy whereby we deliver the genome editing into the body. For patients, for example, for the sickle cell anemia that has been done is being delivered through the stem cells into the bone marrow. Okay, and from the bone marrow, the stem cells, the engineered stem cells, will actually begin to express the kind of hemoglobin that is being desired. Several models also are also coming up. People proposing what they call nanoparticles. Some people are pushing forward for using the usage of nanoparticles for delivery. Some people are also pushing for the inactivated viral particle of delivery into the system. So we have quite a number of mode of delivery to the patient. But in the laboratory, we have you can use electroporation. We can use microinjection. In some microorganisms, you can use heat shock to move your plasmid across the membrane to actually go and put the gene, the engineered gene in the embryo or in the cell that you actually desire the effect that you have actually engineered the gene for.
Thank you, sir. The next one is, sir, what role do bioinformatics and computational tools play in guiding genome editing strategies for cancer treatment?
Bioinformatics plays a lot of role because in designing your guide RNA, you need the bioinformatics tools. In designing the guide RNA, even you have to design some primers during the course because you actually need to ascertain whether your work is well done. In fact, we have some bioinformatics tools that even before getting to the lab, we can even predict. You can have predict possible gel that you can get. You can predict possible sequence that you are going to get using bioinformatics too. So bioinformatics plays a whole lot of role when it comes to your design of most of what you want to use for your genome editing. So that knowledge is very, very important for you to get.
Right, sir. So one last question, sir. How can interdisciplinary collaborations between researchers, clinicians, and industry stakeholders accelerate the translation of genome editing technology into clinical practice for cancer patients?
Okay, is asking the importance. I think it's very, very important. Of course, well, we the scientists cannot do it alone. There must be collaboration across board, from scientists to clinicians. You know, we take it from the preclinical level. We need to collaborate with the clinicians as it's also passed through different processes before going to clinical trials. So at our own end as scientists, we first of all ascertain the basic things we do. You do them in silico using bioinformatics to actually do it. You can run it through your in vitro and then go to in vivo using animal models and then work with clinicians. So it's a whole lot of chain actually for you to have a successful research in this field of anything that has to do with live patient. Of course, you have your pharmacists, your doctors, your clinicians. You need everybody around. All hands actually must be on deck to be able to get a very good result that will be safe, and then that will be able to comply with all the rules and guidelines that govern usage of any therapy that goes into anybody.
Thank you, sir, for answering wonderfully to all the questions. I think the participants may be satisfied with your answers. Now we come to the conclusion of this session. So in conclusion, 40th SPSR International Webinar on adopting genome editing technology in combating cancer disease has been both enlightening and inspiring, shedding lights on the transformative potential of genome editing in the realm of cancer research and therapy. Through comprehensive discussion and in-depth analysis, we have underscored the critical role that genome editing techniques can play in targeting oncogenic mutations, elucidating the underlying mechanism of cancer progression, and developing novel treatment modalities. As we navigate the complexities of cancer biology, it is clear that harnessing the power of genome editing holds the key to unlocking new avenues for precision medicine and personalized therapies tailored to individual patients. However, it is imperative that we continue to address the challenges posed by the off-target effects, delivery methods, and ethical considerations to ensure the responsible and ethical use of these technologies. Moving forward, let us seize this momentum to foster collaboration, drive innovation, and accelerate progress in our collective quest to conquer cancer. Together, we can harness the potential of genome editing to revolutionize cancer care and make meaningful strides towards a future free from burden of this devastating disease. We extend our sincere gratitude to Dr. Shashi Alok, the National President of SPSR, for gracing us with his esteemed presence today. In acknowledging your dedication and leadership, we express our heartfelt thanks for your valuable contribution. In extending our deepest gratitude, we express our sincere appreciation to the esteemed speaker, Dr. Joseph Bamidele Minari, whose generosity in sharing his profound expertise and invaluable perspectives has enriched our understanding of adopting genome editing technology in combating cancer disease. A special acknowledgement is extended to our convener, Mrs. Monika Sabharwal, ma'am, whose pivotal role in orchestrating this insightful event has been instrumental. We extend heartfelt thanks to all the participants, recognizing the collective effort that has effectively pushed the boundaries of knowledge and fostered a spirit of collaborative learning. Together, your contributions have made this webinar a resounding success, and we look forward to continued collaboration in advancing pharmaceutical sciences and research. Thank you for being an integral part of the 40th SPSR International Webinar. Your active participation and contributions have been invaluable, and we look forward to future endeavors that contribute to the continuous betterment of healthcare and well-being for all. Have a wonderful day ahead. Thank you.